Welcome, curious minds, to the deep dive. Today, we're embarking on a crucial journey into the very architecture of life around us, the morphology of flowering plants. If you're tackling your board exams, gearing up for NEET, or simply aiming to build an unshakable foundation in life sciences, you know how indispensable these foundational concepts are. What might seem like mere description is actually the key to decoding nature's intricate language, unlocking complex biological processes, and truly understanding the incredible diversity we see. And joining me, as always, to be our expert guide through this fascinating subject is our brilliant biology teacher. It's wonderful to have you here.
Thank you so much. It's always a pleasure. You've really hit on a very important truth there. Morphology, at its heart, is the study of external structure. And while it might, at first glance, appear to be just descriptive, a list of where it truly forms the bedrock for understanding everything that follows in plant biology. Things like how plants function, that's physiology, how they interact with their environment, ecology, and even how they've changed over millions of years, that's evolutionary biology. For a long time, detailed observation and description were the very essence of biology, or what we used to call natural history. And even today, this foundational knowledge is indispensable, especially for students like you who are preparing for these demanding competitive exams here in India. So our mission today is to systematically unpack these detailed structural features of higher plants, taking you through your study materials step by step, ensuring every single concept is crystal clear. Think of this as, well, your personalized in-depth coaching session.
That's a perfect framing for our deep dive. Our focus today is squarely on structural organization in plants, specifically zooming into morphology of flowering plants, ###my, introduction to plant morphology and historical context. So to really appreciate the science of morphology, let's maybe set the stage a bit. How did this meticulous observation and detailed description become such a cornerstone of biological study, historically speaking?
That's an excellent question, and it gives vital context, really. Imagine the earliest biologists, long before we had powerful microscopes or genetic sequencing. Their work was, in essence, natural history. They were primarily naturalists, driven by curiosity, who focused intensely on observing and meticulously recording the gross structural features of life forms. This involved looking at both external features, things visible to the naked eye, and internal ones, often discerned with just simple magnifying lenses. That whole era was characterized by an astonishing level of descriptive detail, almost like collecting and documenting every single puzzle piece before you even knew what the full picture looked like.
Wait, painstaking work.
Absolutely. Now, some students might initially find such detailed descriptions a bit, tedious perhaps, thinking, why do I need to know every single part of a flower? But it's critical to understand that this descriptive foundation wasn't just an end in itself. It proved absolutely crucial for what came later, what we call reductionist biology, where scientists started focusing more on living processes rather than just static forms. All that rich descriptive data, all those puzzle pieces, they became incredibly meaningful. They helped frame precise research questions in fields like plant physiology,
where we study how plants grow and respond.
Exactly. And in evolutionary biology, where we trace their lineage. So for our purposes today, we'll be describing the structural organization of plants, including the structural basis of their physiological phenomena. And we'll present the plant aspects separately for absolute clarity.
That truly puts the importance of observation into perspective. And speaking of pivotal figures in this field, your study material highlights Catherine Esau. Can you tell us a bit about her immense significance in the field of plant anatomy?
Absolutely. Catherine Esau, born in Ukraine back in 1898, is truly considered a titan in the world of plant biology. Her journey was quite remarkable. She initially pursued her agricultural studies in Russia and Germany, eventually earning her doctorate in the United States in 1931. Her early research was groundbreaking. For example, she did crucial work on understanding how the curly top virus spreads through plants.
Oh, the virus. How did she figure that out?
Well, she specifically identified its movement through the food-conducting phloem tissue. That was a significant insight into plant pathology at the time. But her most profound contribution, the one that really cemented her legacy, came with her book Plant Anatomy, published in 1954.
Plant anatomy. Okay, what made it so special?
It wasn't just another textbook. It adopted what she called a dynamic developmental approach, which completely revolutionized the discipline. Instead of just describing static structures, she showed how these structures develop and function dynamically. It made plant anatomy much more understandable and relevant. Its impact was enormous, literally sparking a revival in plant anatomy worldwide. She followed this with The Anatomy of Seed Plants in 1960. This book was so comprehensive, so encyclopedic in its detail, that it became affectionately known as the Webster's of plant biology.
Wow, like the dictionary for plant structure?
Pretty much. If you wanted to know anything about plant structure, this was the definitive reference. And her immense contributions didn't go unnoticed. She was elected to the National Academy of Sciences in 1957, a tremendous honor, making her only the sixth woman to receive that recognition. And in 1989, she was awarded the National Medal of Science by President George Bush, one of the highest scientific honors in the United States. Peter Raven, a very renowned botanist, famously recalled that even at the age of 99, when she passed away in 1997, Catherine Esau absolutely dominated the field of plant biology. Her dedication to detailed work really provided the foundational language for generations of botanists.
What an incredible legacy. And for all of you preparing for competitive exams, knowing about such key figures, their contributions, and their timelines can really add depth to your answers. Sometimes these historical facts even pop up in general knowledge sections of exams. Now let's shift our focus to the stars of our deep dive. Angiosperms, the flowering plants. They exhibit such a mind-boggling diversity. Yet, despite this wide variation, what are the common structural features that unite all higher plants, specifically angiosperms? How do we even begin to categorize them?
That's a fascinating paradox in biology, isn't it? The sheer diversity in external structure or morphology among angiosperms is truly wide-ranging. Think about it. From a tiny duckweed floating on water to a massive towering banyan tree. Yet despite this enormous variation, all angiosperms share a fundamental, unifying blueprint. They are consistently characterized by the presence of roots, stems, leaves, flowers, and fruits. This common blueprint, even with all the variations, highlights a shared evolutionary heritage.
So there's an underlying pattern despite the variety.
Precisely. And for us to successfully classify and understand any living organism, including these higher plants, it's absolutely essential to use standard technical terms and standard definitions. Imagine trying to describe a car without standard terms like engine or wheel. It would just be chaos. Without this common language, scientific communication would be impossible. And it's not just about finding commonality. We also need to understand the possible variations in these parts. These variations are often remarkable adaptations.
Adaptations? Like how?
Well, adaptations that plants have developed to their specific environments or to perform specialized functions. Think about how plants adapt for survival in different habitats, deserts, water, mountains, or for protection against herbivores, maybe with thorns, or for climbing up supports like vines, or for efficiently storing reserve food material like starch or sugar in roots or stems. Each variation tells a story of adaptation. To help you visualize this basic blueprint, imagine pulling out a common weed from your garden, perhaps a small mustard plant. What do you immediately notice?
Roots in the soil, stem, leaves, maybe flowers.
Exactly. You see the roots anchoring it firmly in the soil. The stem rises above the ground, supporting everything else. Leaves emerge from the stem. And depending on the season, you might also see flowers, and perhaps even small fruits forming. This simple act reveals the fundamental organization of all angiosperms. We clearly distinguish the root system as the underground part, responsible for anchorage and absorption, while the portion above the ground forms the shoot system, responsible for photosynthesis, reproduction, and support. If you refer to Figure 5.1 in your study material, you'll see a clear diagrammatic representation of these two primary systems and their components. This visual is really key to understanding the foundational parts we'll be discussing throughout the session. #2, the root system.
Okay, so let's begin our detailed exploration by going underground, literally, with the root system. Can you walk us through the different types of root systems found in flowering plants? This distinction seems absolutely crucial for identification, especially in exams.
Yes, it definitely is. Let's take this step by step and clearly differentiate the types of roots, focusing on their origin and structure. It's all about where they come from. First, we have the taproot system. This system is characteristic of the majority of dicotyledonous plants, dicots. A classic example is the mustard plant. Its formation begins with the direct elongation of the radical. Remember the radical?
An embryonic root from the seed, right?
Exactly. This radical then grows downwards, usually deep into the soil, forming the main central primary root. From this robust primary root, thinner side branches emerge. We call these lateral roots of several orders. You get secondary roots branching off the primary, then tertiary roots off the secondary, and so on. Together, this strong primary root and all its subsequent branches form the taproot system. If you look at Figure 5.2a in your study material, you'll clearly see that distinct, thick main root with thinner lateral branches extending from it, almost like a miniature tree underground.
Okay, so a main root with branches. That's the taproot, typical for dicots. What's the alternative?
In contrast, we have the fibrous root system. This is typical of most monocotyledonous plants, monocots, with the wheat plant being an excellent example. Here, the primary root, which initially develops from the radicle, is quite short-lived. It quickly degenerates or is replaced within a short period after germination. In its place, a large number of roots emerge. But crucially, and this is the key difference, these roots originate directly from the base of the stem itself, not from the radicle's direct elongation. These numerous, slender, and pretty much uniformly sized roots then spread out, forming a dense cluster. This cluster, resembling a tangled mat, is what we call the fibrous root system. Figure 5.2b in your material vividly shows this dense, bushy cluster of roots originating from the stem base in wheat.
So no single main root, just a bunch from the stem base. Got it. And you mentioned a third type.
Yes, there's a distinct third category, adventitious roots. These are roots that arise from any part of the plant other than the radicle. Their unconventional origin is a key differentiating factor. Think about the incredible diversity here. In grass, roots can emerge from the nodes along the stem. The intriguing monstera plant develops aerial roots that dangle down.
Oh, like those dangly bits on some houseplants.
Exactly. And of course, the iconic banyan tree with its massive prop roots descending from branches, sometimes reaching the ground and forming new trunks. These are all adventitious roots. If you refer to figure 5.2c, you'll see the distinct appearance of these roots arising from unusual locations. Now, for competitive exams, this is a vital point. Always remember to pay close attention to the origin of the roots to avoid confusing fibrous roots with adventitious roots based solely on their appearance. Their developmental pathway is the true differentiator.
That's a remarkably clear distinction between the types of root systems tap, fibrous, and adventitious based on origin. So once these roots are established, what are the primary roles or functions that this root system performs for the plant's overall survival and vigorous growth? Beyond just holding the plant in place, of course.
You're absolutely right. They do much more than just anchorage. The functions of the root system are fundamental to a plant's existence and, yes, very frequently tested in exams. First and foremost, roots are responsible for the absorption of water and essential minerals directly from the soil. This is their most vital life-sustaining role. Secondly, as you mentioned, they provide anchorage and proper mechanical support to the entire plant, firmly holding it in place against forces like strong winds or heavy rain.
Okay, absorption and anchorage. What else?
Thirdly, many roots are actually modified for the storage of reserve food material. Think of common vegetables that we eat, daily carrots and radishes, for instance. These are not just roots, they're actually modified taproots, specifically specialized for storing an abundance of food, like starch or sugar. This helps the plant survive periods of scarcity or fuel its next growth phase.
Ah, like storing energy for later.
Precisely. And finally, roots are also active sites for the synthesis of certain plant growth regulators, or hormones. These hormones, produced in the roots, then travel to other parts of the plant and play crucial roles in regulating overall plant development, growth patterns, and responses to the environment. So they're not passive, they're dynamic, multifunctional organs.
Synthesis of growth regulators, too. Interesting. Now, let's zoom in a little further, right to the very tip of the root. This region seems critical for continuous growth and penetration through the soil. Can you walk us through the distinct regions of a root which are vital for understanding how it grows and functions?
Certainly. This is fundamental to understanding growth. Let's take this step by step, visualizing the root tip as we move along its length, starting from the very end and moving upwards. As I describe each region, you can mentally follow along with figure 5.3 in your study material for a clear diagrammatic representation. It really helps to visualize. Okay, at the very apex, the absolute tip of the root, you'll find the root cap. This is a thimble-like structure, almost like a tiny helmet, covering the delicate growing tip.
A protective cap.
Exactly. Its primary function is protection. As the root pushes its way through the soil, which can be quite abrasive and resistant, the root cap shields the tender apex, preventing mechanical damage to the actively dividing cells within. It's the root's first line of defense. Pause. Now, moving just a few millimeters above the root cap, we encounter the region of meristematic activity. This is where the core of root growth truly begins. The cells in this region are quite distinctive. They are very small, have characteristically thin cell walls, and possess dense protoplasm, which indicates high metabolic activity. Their crucial function is to divide repeatedly through rapid mitosis, constantly producing new cells. This continuous cell division is what adds new cells to the root, contributing to its overall growth.
Pause. So this is the cell factory, basically.
Yeah, that's a good way to put it. Just proximal meaning, slightly further away from the tip to this meristematic region, lies the region of elongation. Here, the cells that were newly formed in the meristematic zone undergo rapid elongation and enlargement. They absorb a lot of water and expand significantly. This increase in cell size and length is primarily responsible for the growth of the root in length. It's this region that truly pushes the root deeper into the soil. Pause.
So the pushing comes from cell stretching, not just division.
That's right, primarily stretching here. And finally, proximal to the region of elongation, we have the region of maturation. In this zone, the cells that have elongated now begin a process called differentiation and maturation. They stop growing in size and instead specialize, developing into various distinct tissue types that make up the mature root, like vascular tissues for transport or ground tissues for support. Crucially, it is from this very region that some of the epidermal cells extend outwards to form extremely fine and delicate thread-like structures known as root hairs.
The root hairs for absorption.
Exactly. These root hairs dramatically increase the surface area of the root, like millions of tiny sponges, which is absolutely essential for the efficient and extensive absorption of water and minerals from the soil.
That's a remarkably clear breakdown. So, if I'm understanding correctly, the meristematic region is the factory generating new cells. The elongation zone is where they stretch and push the root forward, contributing most to length increase. And the maturation zone is where cells specialize and form those vital root hairs for absorption. Understanding these distinct regions is absolutely crucial for any questions on root growth and development in competitive exams. It explains how the root actually does its job. ##tag 3, the stem. That's a powerful understanding of how roots operate underground. Now, shifting our gaze upwards, let's talk about the plant's main support structure above ground, the stem. What are the key features that truly distinguish a stem from a root, and what are its main functions in the plant's life?
Excellent point. The stem is essentially the ascending part of the plant axis, generally growing upwards, often towards sunlight. It's the central scaffold that bears all the other vital aerial parts: branches, leaves, flowers, and fruits. A key distinguishing feature from the root is its developmental origin. The stem develops from the plumule of the embryo in a germinating seed.
Whereas the root came from the radical.
Exactly. Plumule for shoot, radical for root. Another definitive structural marker of a stem, completely absent in roots, is the presence of distinct nodes and internodes. Think of them like the segments of a bamboo stick, perhaps. Nodes are the specific regions on the stem where leaves and often branches are born. Internodes are simply the portions of the stem located between two consecutive nodes.
Nodes where things attach, internodes are the spaces between. Clear.
Right. Stems also consistently bear buds. These can be terminal buds located at the very apex of the stem, responsible for primary growth and length, or axillary buds found in the axil, that's the angle between a leaf and the stem, which have the potential to develop into branches or flowers. Visually, young stems are typically green, indicating they can do some photosynthesis, but as they age, especially in woody plants, they often become darker, woody, and turn brown.
Okay, so nodes, internodes, buds coming from the plumule, and its functions.
Its primary role is support and display. It effectively spreads out branches bearing leaves, maximizing their exposure to sunlight for efficient photosynthesis. It displays flowers prominently, which is crucial for attracting pollinators and ensuring reproduction. And it supports fruits, essential for seed dispersal. Beyond support, the stem is a highly efficient conduit for conduction. It's like the plant's plumbing system.
Transporting stuff up and down.
Exactly. It transports water and dissolved minerals upwards from the roots to all aerial parts, and simultaneously, it conducts photosynthase, the food produced in the leaves, to all other parts of the plant, both upwards and downwards, wherever they are needed for growth or storage. And it's important to note that stems, much like roots, can also be remarkably modified to perform specialized functions.
More adaptations. Like what?
Well, for instance, the underground stem of a potato is a tuber, modified specifically for the storage of food. If you've ever seen grapevines or passion flowers, their delicate tendrils are actually modified stems, providing support by coiling around objects, allowing the plant to climb. Thorns in plants like citrus or bougainvillea are hardened, modified stems offering protection against herbivores. And finally, some stems, like the rhizome of ginger or the stolons of strawberries, are modified for vegetative propagation, allowing the plant to reproduce asexually and spread rapidly.
Wow, stems can be quite versatile. So being able to list these features, functions, and especially the examples of modifications is clearly very important for exams. Hashtag hatch, tag 5. The leaf, absolutely. Now, the leaf, undoubtedly one of the most vital parts of a plant, truly the powerhouse of photosynthesis, where the plant harnesses the sun's energy. Could you elaborate on its general features, its intricate structure, and the fascinating patterns it displays?
You're absolutely right. The leaf is central to a plant's very existence. Generally, a leaf is a lateral, typically flattened structure designed perfectly to capture maximum sunlight. It is born on the stem, developing precisely at a node. A significant diagnostic feature is that it consistently bears a bud in its axil. Remember, the angle formed between the leaf and the stem. This axillary bud holds the potential to later develop into a branch or perhaps a flower.
So the bud in the axil is a key leaf feature.
It is. In terms of their origin and arrangement, leaves originate from the shoot apical meristems, those actively growing tips of the stem, and they are arranged in an acropital order.
Acropital. That means youngest at the top, oldest at the bottom, right? Like we saw with racemose inflorescences.
Exactly the same principle. Youngest leaves near the apex, older ones further down. Functionally, due to their characteristic green color from chlorophyll and their broad, flattened structure, leaves are the most important vegetative organs for photosynthesis. That incredible process where light energy is converted into chemical energy, creating the plant's food.
Okay, let's take an even closer look at a typical leaf. What are its distinct parts and what role does each play?
A typical leaf, as you can visualize in figure 5.4a of your study material, consists of three main parts, each with a specialized role. Let's break them down. First, we have the leaf base. This is the part by which the leaf is attached directly to the stem. It's the point of connection. Interestingly, the leaf base may sometimes bear two small lateral leaf-like structures called stipules. While not all leaves have them, their presence or absence is a key identifying feature for certain plant groups.
Stipules. Okay, small leaf-like things at the base.
Correct. In monocotyledonous plants, like grasses, a common adaptation is for the leaf base to expand into a sheath, which partially or even wholly covers the stem, providing additional support and protection. And in some leguminous plants, such as the pea plant, the leaf base can become distinctly swollen, forming a structure known as a pollenus. This pulvinus is quite remarkable because it plays a crucial role in leaf movements, like the sleep movements you see in some plants where leaves fold up at night.
A swollen base for movement.
Fascinating.
What's next after the base?
Next is the petiole. This is simply the stalk of the leaf, connecting the leaf blade to the stem. Its primary function is quite clever. It helps hold the leaf blade, or lamina, up to the light, allowing it to orient itself optimally to capture maximum sunlight for photosynthesis. A key benefit of having long, thin, and flexible petioles, as seen in the peepal tree, for example, is that they allow the leaf blades to flutter freely in the wind.
Why would fluttering be useful?
It helps in two crucial ways. It cools the leaf surface, preventing overheating, especially on hot days, and it brings a constant supply of fresh air, rich in carbon dioxide, right to the leaf surface, thereby enhancing the rate of photosynthesis. It's like nature's built-in air conditioner and CO2 delivery system.
Clever design. And the main part of the leaf.
Finally, we have the lamina, also widely known as the leaf blade. This is the green, expanded, and typically the most prominent part of the leaf. It's crisscrossed by a network of veins and veinlets, almost like a miniature road map. Typically, there's a prominent central vein running through the lamina called the midrib. The veins themselves serve dual functions. They provide crucial structural rigidity to the leaf blade, acting like a skeletal framework that prevents it from wilting or tearing easily. And they also function as highly efficient channels for the transport of water and essential minerals to all parts of the leaf, and for the transport of manufactured food materials, or photosynthetes, away from the leaf to other parts of the plant. It's important to remember that the lamina exhibits wide variation across different plant species in terms of its overall shape. The nature of its margin, smooth, toothed, lobed, the shape of its apex, its surface texture, hairy, smooth, and the extent of its incision. These variations are often key features for plant identification.
Right, the lamina's details are key for ID. And you mentioned veins. The arrangement of veins within the lamina, the venation, is another key characteristic, isn't it? What are the main types?
Indeed, venation refers specifically to the pattern of arrangement of veins and veinlets within the lamina. This pattern isn't just for looks, it's a fundamental classification trait. There are two primary types you absolutely need to know. First, we have reticulate venation. In this type, the veinlets form an intricate network-like pattern throughout the leaf blade, almost like a finely woven mesh or a spider web. This type of venation is characteristic of most dicotyledonous plants. If you pick up a peepal leaf or a rose leaf, you can clearly visualize this by holding it up to the light. You can also see this clearly depicted in Figure 5.4b in your study material.
Reticulate means network-like for dicots, okay?
Second, we have parallel venation. Here, the veins run parallel to each other within the lamina, without forming a distinct interconnected network. They can run parallel to the midrib, like in many grasses, or they can run parallel to each other from the base of the lamina, converging towards the apex. This type of venation is characteristic of most monocotyledons. Think of a banana leaf or a grass blade. You'll notice the prominent lines running side by side without crossing over extensively. Figure 5.4c will help you visualize these parallel lines.
Parallel lines for monocots. Got it.
For competitive exams, this is a classic distinguishing feature between dicots and monocots, a real fundamental concept, and is very frequently tested. So definitely commit these two types and their examples to memory.
Absolutely. Now, besides a venation, leaves are also categorized as simple or compound. This can sometimes be a bit confusing for students. How do we differentiate between them definitively? What's the key thing to look for?
That's a very important distinction. And you're right, it can be confusing. It hinges on two main things: the extent of the lamina's incision and, crucially, the presence or absence of that axillary bud we talked about. Let's break it down carefully. A leaf is considered a simple leaf if its lamina is either completely entire, meaning it's undivided, or if it is incised, the incisions do not reach or touch the midrib. Even if the leaf margin is deeply lobed, like in an oak leaf, as long as the divisions don't completely separate the lamina into distinct parts that reach the midrib, it remains a simple leaf. And here's a key diagnostic feature for your exams. A bud is always present in the axil of the petiole of a simple leaf.
Okay, simple leaf lamina not divided to the midrib and has a bud in its axil. What about compound?
In contrast, a leaf is categorized as a compound leaf when the incisions of the lamina are so deep that they reach right up to the midrib, effectively breaking the lamina into a number of separate units called leaflets. Now, here's the crucial diagnostic feature, the secret code for competitive exams. While a bud is present at the axil of the petiole of the entire compound leaf at the base where it joins the stem, there will be no bud in the axil of the individual leaflets.
Ah, that's the key differentiator. No buds at the base of the little leaflets.
Exactly. This helps you definitively distinguish a true compound leaf from, say, a small branch bearing multiple simple leaves. It's a common trick question in exams. Compound leaves themselves are further divided into two main types, which you can visualize easily with Figure 5.5 in your study material. First, the pinnately compound leaf. In this type, a number of leaflets are arranged laterally on a common axis. This common axis is called the rhoches, and the rhoches itself actually represents the midrib of the original simple leaf. A classic example is the neem tree, where you see many small leaflets arranged neatly along that central stalk, the rhoches, almost like the barbs of a feather. Figure 5.5a shows this clearly.
Pinnate like a feather, leaflets along a central rhoches. Okay.
Second, the palmately compound leaf. Here, the leaflets are not arranged along the central rhoches. Instead, all the leaflets are attached at a common point, specifically at the tip of the petiole, radiating outwards like the fingers of your palm. An excellent example is the silk cotton tree, which has those broad leaflets fanning out from a single point. You can see this distinct arrangement in Figure 5.5b.
Palmate, like a palm, leaflets radiating from one point. Got it. That distinction about the axillary bud is incredibly helpful and, as you said, so vital for competitive exams to classify correctly. Now let's talk about phyllotaxy. The pattern of leaf arrangement on a stem is often unique to a species and aesthetically quite fascinating. Can you explain the main types of phyllotaxy and maybe why this arrangement matters biologically?
Phyllotaxy is precisely that. It refers to the specific pattern of arrangement of leaves on the stem or a branch. This arrangement isn't random at all. It's highly organized. It's really an evolutionary strategy designed to maximize light exposure for each leaf, ensuring efficient photosynthesis and minimizing shading of lower leaves by upper leaves. There are typically three main types, which you can visualize in Figure 5.6 of your study material. First, the alternate type. In this arrangement, a single leaf arises at each node, and these leaves are arranged in an alternate or spiral manner along the stem. If you were to look down on the stem from above, you'd see the leaves spiraling around it, ensuring each gets its share of sunlight. Common examples include China rose, mustard, and sunflower plants. Refer to Figure 5.6a to see this single leaf per node alternating pattern.
One leaf per node spiraling up. Alternate. Makes sense.
Second, the opposite type. Here, a pair of leaves arises at each node, and these two leaves lie directly opposite to each other across the stem. Excellent examples are calotropis and guava plants. Figure 5.6b clearly shows two leaves emerging directly opposite each other at the same node.
2 leaves per node, directly across from each other. Opposite.
And third, the whorled type. This is the arrangement where more than 2 leaves arise at a single node and form a whorl, meaning they form a circle or ring around the stem at that point. This pattern is less common but very distinctive when you see it. A classic example is Alstonia, which is beautifully illustrated in figure 5.6c, showing multiple leaves radiating from a single point on the stem.
More than 2 leaves at a node in a circle world. Okay.
For exam relevance, knowing these definitions and their specific plant examples for each type, alternate, opposite, whorled, is very important for multiple choice questions. These simple patterns are key to identifying a plant just by looking at how its leaves are arranged. ## V, the inflorescence.
We often think of flowers individually as beautiful standalone units, but you mentioned their arrangement on the plant is also systematically classified. What exactly is an inflorescence, and what are its major types?
That's an excellent point and a fundamental concept in floral morphology. A flower, at its most fundamental level, is actually a modified shoot.
A modified shoot. How so?
Think about it. What normally grows into stem and leaves, the shoot apical meristem, changes its developmental program and transforms into a floral meristem. During this remarkable transformation, the internodes of that shoot do not elongate, and the axis itself becomes highly condensed. Instead of producing leaves, this modified apex then produces different kinds of floral appendages laterally at successive nodes, sepals, petals, stamens, carpels. Now, when a shoot tip completely transforms into a single solitary flower, well, that's just called a solitary flower. However, when flowers are produced in a specific cluster or arrangement on the floral axis, that entire arrangement is termed the inflorescence. It's basically the way flowers are presented or grouped on the plant.
So an inflorescence is a group or arrangement of flowers on an axis. What are the main ways they can be arranged?
The two major types of inflorescences are defined by a crucial difference: whether the main axis of the inflorescence develops into a flower and stops growing, or if it continues to grow indefinitely. Let's look at them sequentially, and I encourage you to visualize with figures 5.7 and 5.8 in your study material as I describe them. First, we have the racemose type of inflorescence. In a racemose inflorescence, the main axis has the capacity to continue to grow indefinitely. It doesn't terminate in a flower, it just keeps on elongating at the tip. Because of this continuous growth, the flowers are borne laterally along the main axis in a specific pattern called acropital succession.
Acropital succession.
Mm-hmm.
We heard that with leaves. Youngest at the top.
Exactly. Think of it like stacking coins. The newest coins are always added on top. So the younger flowers are located towards the apex or the very tip of the inflorescence, while the older, more mature flowers are found towards the base. New flowers are constantly forming at the top. Refer to figure 5.7. Imagine that growing stalk with younger, smaller flowers appearing at the very top. For an exam tip, think A for acropital and A for apex. Youngest flowers are always at the apex.
Racemose. Axis keeps growing. Youngest flowers at the apex. Acropital. Okay. What's the other type?
Second, in stark contrast, we have the cymose type of inflorescence. In the cymose inflorescence, the main axis terminates in a flower. This very act limits its growth. Once the apex becomes a flower, it can't grow any further in that direction. Because the main axis ends in a flower, the flowers are borne in the basipetal order.
Basipetal, so opposite of acropital.
Precisely. Basipetal order signifies that the oldest flower is located at the apex, the one that terminated the main axis, and the younger flowers develop below it or towards the base of the inflorescence, often on lateral branches that arise below the terminal flower. So the oldest flower is always at the top, and newer ones branch out from beneath. Visualize figure 5.8. You'll see a prominent mature flower at the very top with newer younger flowers branching out from below it. For your exam preparation, think B for basipetal and B for basipesh. The youngest flowers are towards the base, developing below the terminal one.
Cymose. Axis ends in a flower, oldest flower at the apex, younger ones below. Basipetal. Got it. That distinction between acropital and basipetal succession is indeed a key identifier for these two types, and probably a common exam question. #tag checks the flower. Absolutely. Now, the flower itself, the reproductive marvel of angiosperms, teeming with intricate details and a breathtaking diversity of forms. Let's dissect its structure and the various ways we classify it.
Yes, let's. And just to reiterate, the flower is the exclusive reproductive unit in angiosperms, precisely designed and evolved solely for sexual reproduction. It's where the magic happens, so to speak. Now, 6.2 parts of a typical flower. The whorls of life. A typical flower is characterized by having 4 distinct kinds of whorls. These are arranged successively in concentric circles on the swollen end of the flower stalk. And that swollen end, the base where everything sits, is called the thalamus or receptacle. Let's refer to figure 5.10 in your study material to visualize these whorls, moving from the outermost to the innermost. The outermost whorl is the calyx. This is composed of individual units called sepals, usually green and protective. Moving inwards, we find the corolla, made up of petals, usually colorful and attractive. Next is the androecium. This is the male reproductive part consisting of stamens. And finally, at the very center is the gynoecium, the female reproductive part composed of carpels.
Calyx, corolla, androecium, gynoecium. 4 whorls outside to inside, CCAG.
Perfect mnemonic. Now, we categorize these whorls based on their primary function. The calyx and corolla are considered accessory organs. Their primary roles are protection, especially in the bud stage, and attraction of pollinators, which is crucial for reproduction. They help, but aren't directly reproductive. The androecium and gynoecium, however, are the reproductive organs, as they are directly involved in the actual process of sexual reproduction, producing pollen and ovules. Now, a special case. In some flowers, like the lily, the calyx and corolla are not clearly distinct. You can't easily differentiate sepals from petals. They look very similar. In such cases, they are collectively termed the perianth.
Perianth when sepals and petals look alike.
Okay. Flowers are also classified by their sexuality. If a flower possesses both the androecium, male part, and the gynoecium, female part, in the same flower, it is referred to as mydasexual. Most flowers are. If a flower has only stamens and is therefore male, or only carpels and is therefore female, it is called unisexual.
Bisexual or unisexual? Simple enough. Now, floral symmetry is another fundamental characteristic, almost like a plant's unique blueprint. Can you explain actinomorphic, zygomorphic, and asymmetric flowers with examples? That sounds like a frequent question area.
It certainly is. Floral symmetry defines how a flower can be divided into equal halves, which is vital for classification and often indicates how it interacts with its specific pollinators. Let's break them down. First, actinomorphic. Radial symmetry. A flower is said to be actinomorphic when it can be divided into 2 equal radial halves by any radial plane that passes through its center.
Like cutting a pizza you said earlier.
Exactly. No matter where you slice through the center, you'll always get 2 equal halves. This implies that the flower parts are arranged symmetrically around a central axis, like spokes on a wheel. Classic examples include mustard, datura, and chili. These flowers often attract a wide range of pollinators because they are accessible from many angles. Second, zygomorphic, bilateral symmetry. In contrast, a flower is zygomorphic when it can be divided into 2 similar halves by only one particular vertical plane. This is much like the symmetry of our own bodies. You can only cut it down the middle along a specific plane to get 2 mirrored halves. This often leads to a more specialized interaction with pollinators, as the unique shape guides the pollinator to a specific landing spot to access nectar and transfer pollen effectively. Examples of zygomorphic flowers include pea, gulmohur, bean, and cassia.
So only one way to cut it in half equally, like our faces.
Good analogy. And finally, asymmetric, irregular. A flower is asymmetric or irregular if it cannot be divided into 2 similar halves by any vertical plane passing through its center. There's simply no plane of symmetry whatsoever. The parts are irregularly arranged. The classic example provided in your study material is canna.
No symmetry at all. Irregular. Got it.
Knowing these definitions, actinomorphic, zygomorphic, asymmetric, and their specific examples, is crucial for competitive exams. They test not just your knowledge, but your ability to visualize and classify based on shape.
And flowers are also classified by the number of their floral appendages, correct? Like in multiples of 3, 4, or 5.
Yes, that's another important layer of classification, especially useful for distinguishing major groups like monocots and dicots. Flowers are categorized based on the number of their floral appendages, meaning the sepals, petals, or stamens, appearing in multiples within a whorl. If the floral appendages are in multiples of 3, the flower is trimerous. You commonly see this in monocots, like lilies. If there are multiples of 4, it's tetramerous. Mustard is an example. And if there are multiples of 5, the flower is pentamerous. This is very common in dicots, like hibiscus, China rose, or roses.
Trimerous 3s, monocots, tetramerous 4s, pentamerous 5s, dicots. Good rule of thumb.
One more quick but important point for classification. The presence or absence of bracts, remember those. Reduced small leaf-like structures found at the base of the pedicel, the flower stalk. Flowers with bracts are called bracteate, and those without them are termed a bracteate. Again, the small detail that can be a big clue in identification.
Bracteate or abracteate. Okay. Now, the position of the ovary relative to other floral parts on the thalamus, this seems like a really key classification that determines a lot about the flower structure and even the fruit later on. Could you elaborate on hypogynous, perigynous, and epigynous flowers and how the ovary position is described in each?
This classification is exceptionally important and frequently tested, as you suspect. It directly reflects how the flower's structure develops around its reproductive core, the ovary. It's all based on the relative position of the calyx, corolla, and androecium in relation to the ovary on the thalamus. Let's go through each type slowly, visualizing with figure 5.9 of your study material. First, the hypogynous flower. In a hypogynous flower, the gynoecium, or the ovary, occupies the highest position on the thalamus. It's essentially sitting on top. All the other floral parts, the sepals, petals, and stamens, are situated below the ovary. Because of this elevated position, the ovary in such flowers is described as superior.
Hypopo means below, so other parts are below the ovary. Ovary is superior. Makes sense. Examples.
Good examples include mustard, China rose, and brinjal. Figure 5.9a clearly depicts this arrangement, with the ovary prominently placed at the top. Second, the perigynous flower. Here, the gynoecium is situated in the center of the thalamus. The fascinating aspect is that the thalamus itself grows up slightly to form a cup-like structure, sometimes called a hypanthium, around the ovary, but the ovary isn't fully enclosed by it or fused to it. The other floral parts, sepals, petals, stamens, are located on the rim of this cup-shaped thalamus, almost at the same level as the ovary.
So everything's kind of around the middle.
Exactly. The ovary in a perigynous flower is said to be half inferior. Think of peri, meaning around. The other parts are around the ovary, forming a ring on the rim of the thalamus. Examples are plum, rose, and peach, as you can see in figure 5.9b and 5.9c. And third, the epigynous flower. In the epigynous flower, the margin of the thalamus grows upward, completely enclosing the ovary and actually becoming fused with its wall. Consequently, all the other parts of the flower, the sepals, petals, and stamens, arise from the top of this structure above the ovary.
So the ovary looks like it's sunk down below everything else.
Precisely. Because the ovary is fully enclosed by the fused thalamus, it appears to be below the attachment point of the other floral parts. Hence, the ovary in such flowers is described as inferior. Think of epi, meaning upon or above. The other parts are upon or above the ovary. Good examples include flowers of guava and cucumber, and also the ray florets of a sunflower. Figure 5.9, though, visually captures this superior placement of other floral parts relative to the inferior ovary.
Hypogynous, superior ovary. Perigynous, half inferior ovary. Epigynous, inferior ovary. Okay. That understanding of ovary position is absolutely vital for comprehending floral formulae and the detailed descriptions of plant families, especially for exam success. Now, let's go into even more granular detail, dissecting each of the 4 floral whorls. What intricate details do we find within the calyx, corolla, androecium, and gynoecium?
Excellent. Let's do that. Each flower normally has these 4 distinct floral whorls, as we saw in the general arrangement in figure 5.10. Now, let's examine each one in detail, focusing on their structure, function, and key variations that are important for identification. First, the calyx. As we said, it's the outermost whorl. Its individual members are called sepals. Generally, sepals are green and have a leaf-like appearance.
And their main job is protection.
Primarily, yes. They protect the delicate inner parts of the flower when it's still in the bud stage, acting like a protective casing. Now, in terms of their cohesion, meaning how they relate to each other, sepals can either be gamosepalous.
Gamo means united.
Correct. Gamosepalous means the sepals are united or fused together, forming a tube or cup. Or they could be polysepalous, where poly means many or free, so the sepals are free and separate from each other. Pause. Second, the corolla. Moving inward, the corolla is composed of petals. Petals are typically the most visually striking part of a flower, usually brightly colored and often fragrant.
And their function is attraction.
Primarily, yes. To attract insects, birds, or other animals for pollination, a vital step in plant reproduction. Similar to the calyx, petals can also exhibit cohesion. They can be gamopetalous if their petals are united or fused, like in a bell-shaped flower, or polypetalous if their petals are free and separate, like in a rose. The shape and color of the corolla vary tremendously across different plant species. It can be tubular, bell-shaped, funnel-shaped, or even wheel-shaped, each shape often an adaptation for a specific pollinator. Now, a very important term for competitive exams related to the corolla and also the calyx is aestivation.
Aestivation, that sounds important. What is it exactly?
It is. Aestivation refers to the mode of arrangement of sepals or petals in the floral bud with respect to the other members of the same whorl. Think of it as how the petals or sepals are folded or overlap before the flower fully opens. It's about their arrangement relative to each other in the bud stage. Refer to Figure 5.11 for visual aid as I describe the main types. This is a very common exam topic. First, valvate aestivation, Figure 5.11a. In this type, the sepals or petals in a whorl simply touch one another at their margins, without any overlapping. Imagine them meeting precisely edge to edge. A clear example is calotropis. Second, twisted aestivation, Figure 5.11b. Here, one margin of an appendage, sepal or petal, consistently overlaps the margin of the next one, and so on, in a regular specific direction, creating a twisted or spiral appearance in the bud. Think of it like a pinwheel or a tightly wound fan. Examples include China rose, lady's finger, okra, and cotton.
So a regular overlap in one direction, twisted.
Yes. Third, imbricate aestivation, Figure 5.11c. This is where the margins of sepals or petals overlap one another, but crucially, not in any particular regular direction. The overlap is irregular and asymmetrical. Some margins might be outside, some inside. It looks a bit more random compared to twisted. Classic examples are Acacia and Gulmohur. Fourth, and quite unique and specific, is vexillary aestivation, also known as papilionaceous aestivation, Figure 5.11d. This specific type is characteristic of pea and bean flowers, common in the family Fabaceae, the legume family.
Vexillary or papilionaceous, what does that involve?
It involves 5 distinct petals arranged in a very specific way. The largest petal, positioned at the back posterior, is called the standard or vexillum. This standard petal conspicuously overlaps the 2 lateral petals, which are called wings. In turn, these 2 wings overlap the 2 smallest anterior petals, which are often fused together to form a boat-shaped structure called the keel. This keel usually encloses the stamens and pistil.
Standard overlaps wings, wings overlap keel. Very specific.
Very specific, yes. Example, pea and bean flowers. Knowing these 4 types of aestivation, valvate, twisted, imbricate, and vexillary in their examples, is absolutely essential for exams. Pause. Okay, third whorl. The androecium. This is the male reproductive organ of the flower, composed of individual units called stamens. Each stamen, in turn, typically consists of 2 main parts: a long, slender stalk or filament, and an anther at its tip. Each anther is usually bilobed, and each lobe typically contains 2 chambers called pollen sacs. It is inside these pollen sacs that the pollen grains, which contain the male gametes, are produced. Interestingly, sometimes a stamen might be present but sterile, meaning it doesn't produce viable pollen. Such a sterile stamen is termed a staminode.
A sterile stamen is a staminode.
Okay. Now, stamens can also show interesting attachments to other floral parts, which gives us more terminology. If stamens are attached directly to the petals, they are called epipetalous. This is clearly seen in bridal flowers. If they are detached to the perianth, when calyx and corolla are undifferentiated, they are termed epiphyllous, common in flowers like the lily.
Epipetalous on petals, epiphyllous on perianth. Got it.
Furthermore, the stamens can show varying degrees of union among themselves within the same whorl. This is called cohesion. If stamens remain completely free and separate from each other, they are described as polyandrous. If they are united into a single bunch or bundle by their filaments, as in China rose, they are monoadelphous, mono meaning one, adelphous meaning brother or group. If they are united into 2 distinct bundles, which is typical of pea flowers, often 9 fused plus 1 free, they are diadelphous. And if they are united into more than 2 bundles, as found in citrus, they are polyadelphous.
Monoadelphous, 1 bundle. Diadelphous, 2 bundles. Polydelphous, more than 2 bundles, based on filament measure.
Correct. It's also worth noting that within a single flower, there can be variation in the length of the filaments. For example, in plants like salvia and mustard, you might observe stamens of different lengths, which can be an adaptation for specific pollination mechanisms. Pause. And finally, the 4th and innermost whorl, the gynoecium. This is the female reproductive part of the flower, and can be made up of 1 or more carpels. The carpel is the fundamental unit. Each individual carpel, or a fused structure of multiple carpels called a pistil, typically consists of 3 crucial parts. First, the ovary, which is the enlarged basal part. This is where the ovules are housed and protected. Resting on the ovary is the style, an elongated tube that connects the ovary to the stigma. It acts as a pathway for pollen tubes to reach the ovules. Finally, at the very tip of the style is the stigma. This is typically the receptive surface. It's often sticky or feathery, specially adapted for capturing and receiving pollen grains during pollination.
Ovary, style, stigma, the 3 parts of the carpal pistil.
Exactly. Inside the ovary, you'll find 1 or more ovules. These ovules are crucial as they contain the female gametes, egg cells. These ovules are attached to a flattened, cushion-like tissue within the ovary called the placenta. Now, the carpels themselves can also show cohesion, just like sepals, petals, and stamens. If a flower has more than one carpel, and these carpels remain free and separate from each other, the condition is called apocarpus, apo meaning away or separate. This is seen in flowers like lotus and rose, where you can see multiple distinct pistils. If, however, the carpels are fused together, forming a single compound pistil with a single ovary, though it might have multiple chambers, the condition is termed syncarpus. Syn meaning together. This is much more common, found in mustard and tomato, for example.
Apocarpus, free carpels. Syncarpus, fused carpels.
Right, and a critical point to remember, especially for exams regarding the life cycle, is the post-fertilization development. After successful fertilization, the ovules inside the ovary develop into seeds, and the entire ovary matures and develops into a fruit. Ovule becomes seed, ovary becomes fruit. Now let's discuss placentation. This refers to the specific arrangement of ovules within the ovary attached to the placenta. This is another crucial feature for identification and classification in your exams. Refer to Figure 5.12, which shows the different types. Let's visualize each one carefully. First, marginal placentation, Figure 5.12a. In this type, the placenta forms a ridge along the ventral suture, the seam, basically, of the ovary. The ovules are borne on this ridge, typically forming two distinct rows. This is characteristic of a simple ovary derived from a single carpel. A perfect example is the pea pod. When you open it, you see the seeds, developed ovules, lined up neatly in two rows along that seam. That seam is where the placenta is. Second, axial placentation, Figure 5.12b. Here, the placenta is axial, meaning it's located along the central axis of the ovary. The ovules are attached to this central axis within a multilocular ovary, an ovary that has multiple chambers or locules formed by partitions called septa. Think of slicing a tomato or lemon or an orange cross-section. You see the ovules attached to the central column in each distinct chamber. Examples include china, rose, tomato, and lemon.
Marginal like a pea pod seam, axial like the center of a tomato or lemon.
Okay.
Third, parietal placentation, Figure 5.12c. In this case, the ovules develop on the inner wall of the ovary or on the peripheral part. The ovary is typically one-chambered initially, but it often becomes two-chambered due to the formation of a false septum, a partition that isn't a true carpellary wall, but an outgrowth that divides the cavity. Examples are mustard and argemone. Fourth, free central placentation, Figure 5.12pc. Here, the ovules are borne on a central axis, similar to axial, but critically, septa partitions are absent. This means the ovary is single-chambered, unilocular, with a central column bearing the ovules that is not connected to the ovary wall by any partitions. Examples include dianthus and primrose.
So central column, but no walls separating chambers, free central.
Exactly. And fifth, basal placentation, Figure 5.1t. This is perhaps the simplest type. The placenta develops right at the base of the ovary, and typically a single ovule is attached to it. Examples include sunflower and marigold, where you find one seed developing at the base of each individual flower, florets at the head. Marginal, axial, parietal, free central, basal. Understanding these varied placentation types is absolutely essential for distinguishing plant families and is a very common area for exam questions. It truly shows the incredible diversity in nature's internal design. ###7, the fruit.
Right. After the flower's remarkable reproductive role, the fruit takes center stage. It's often a delicious reward for us and clearly a clever dispersal mechanism for the plant. What precisely defines a fruit, botanically speaking, and what are its key components?
You're right, the fruit is a defining characteristic feature of flowering plants, exclusive to angiosperms. Scientifically, it is defined as a mature or ripened ovary, which develops specifically after fertilization. This means the process of fertilization triggers hormonal changes that cause the ovary wall to swell and transform, protecting the developing seeds inside. Now, there's an interesting exception to this rule. If a fruit forms without the fertilization of the ovary, it's called a parthenocarpic fruit.
Parthenocarpic. Seedless fruits? Often, yes. The most common example of a naturally occurring parthenocarpic fruit that you're very familiar with is the banana, which is typically seedless because fertilization doesn't occur properly or isn't needed for fruit development.
Ah, okay. So a normal fruit develops from a fertilized ovary. What's its structure like?
Structurally, a typical fruit generally consists of two main parts: an outer wall, which is called the pericarp, and one or more seeds enclosed within. The pericarp itself is derived from the ovary wall. The pericarp can be either dry, as in nuts or grains or pods like peas, or it can be fleshy, as in mangoes, tomatoes, or berries. When the pericarp is thick and fleshy, it's typically differentiated into three distinct layers, each with a specific role. The outermost layer is the epicarp. This is essentially the skin or rind of the fruit. Think of the thin outer peel of a mango or a grape. The middle layer is the mesocarp. This is often the fleshy, juicy, edible part in many fruits. In a mango, this is the yellow, sweet pulp that we love to eat. In a peach, it's the fleshy part. And the innermost layer is the endocarp. This layer can vary greatly. It might be hard and stony, like the pit of a peach or a plum, or the stone of a mango, which encloses the seed. Or it can be leathery or papery, like the core of an apple, or even juicy, like in citrus fruits, where it forms the pulp segments.
Epicarp, skin; mesocarp, flesh; endocarp, inner layer, maybe stony.
Got it. Exactly. Your study material highlights the drupe as a common fruit type, and figure 5.33 is excellent for illustrating this. Drupes are characterized by developing from a monocarpellary superior ovary, meaning formed from a single carpel, and the ovary was superior, and they typically contain only one seed. Let's look at two classic examples of drupes to understand the pericarp layers clearly, as shown in the figure. In a mango, figure 5.33a, the pericarp is beautifully differentiated. You have the outer thin epicarp, the skin, the middle fleshy and edible mesocarp, the pulpoid, and then the inner stony hard endocarp that encloses the actual seed inside. What we often casually call the seed in a mango is, botanically speaking, the hard endocarp containing the true seed within it.
So the hard bit is the endocarp, not the seed itself. Interesting. What about coconut?
Now consider a coconut, figure 5.13b. This is also botanically classified as a drupe, surprisingly to many, but it has a significant modification compared to the mango. While it also has an epicarp, the smooth outer green or brown layer, and a hard endocarp, the shell enclosing the white meat, its mesocarp is distinctly fibrous. This fibrous mesocarp is what we commonly know as the core, which is used for ropes, mats, and stuffing. The white edible part and the coconut water are actually the endosperm of the seed inside the endocarp.
Wow, so the coconut's fibrous husk is the mesocarp. That's quite different from mango.
It is. This fibrous, air-filled mesocarp makes the coconut a unique example of a drupe with a specialized pericarp, perfectly adapted for buoyancy and dispersal by water over long distances. These examples perfectly illustrate how the pericarp layers can vary and adapt, even within the same basic fruit type, drupe, to serve different functions, especially in seed dispersal. Hashtag HHA.
Following the fruit, we arrive at the seed, truly the plant's entire future encapsulated. A tiny package of potential, ready for dispersal. What exactly is a seed, botanically speaking, and how do dicotyledonous and monocotyledonous seeds differ in their fundamental structure? This seems like a very important comparison for exams.
It absolutely is. The seed is the culmination of sexual reproduction in flowering plants, the very beginning of the next generation. Scientifically, remember, the ovules, located within the ovary, develop into seeds after successful fertilization. Fundamentally, a seed is composed of two main parts: a protective outer covering called the seed coat, and a miniature dormant plant called an embryo. The embryo itself is a complete, albeit tiny, blueprint of the future plant. It consists of a radicle, which will become the embryonic root, an embryonal axis, the central stalk connecting the parts, and either one or two cotyledons, which are embryonic or seed leaves. Let's first examine a typical 8.2 structure of a dicotyledonous seed, using common examples like gram chana or pea matar. Please refer to figure 5.44 in your study material to follow along with the parts. It helps to visualize. Okay, the outermost covering is the seed coat. This protective layer actually has two sublayers: an outer, tougher layer called the testa, and an inner, thinner, more delicate layer called the tegmen. These layers shield the precious embryo inside. Correct. On the seed coat, you'll usually notice a prominent scar called the hilum. This hilum marks the exact point where the developing seed was attached to the fruit wall, or the placenta, within the ovary. It's like the seed's belly button, its former point of connection. Just above the hilum, there's usually a small pore called the micropyle. This tiny opening is actually a remnant from the ovule stage, and it's crucial. It facilitates the absorption of water into the seed during germination, which is often the first step to awakening the embryo. It also allows for gaseous exchange.
Hilum, scar, micropyle, pore for water gas. Okay, what's inside?
Inside this protective seed coat is the embryo. The embryo consists of an embryonal axis and, characteristic of dicots, two cotyledons. These cotyledons in many dicots, like gram or pea, are often quite fleshy and are typically packed full of reserve food materials, proteins, starches, or fats. This stored food provides crucial nourishment to the developing seedling during germination, before its own leaves emerge and it can start photosynthesizing. This is why pulses like gram and pea are so nutritious for us. We're eating the stored food from the cotyledons.
So the cotyledons are like the baby plant's lunchbox.
An excellent analogy. The embryo also has, at one end of the embryonal axis, the radicle, which is the embryonic root. Upon germination, this radicle will be the first part to emerge and grow downwards, anchoring the seedling and absorbing water. And at the other end of the embryonal axis is the plumule, which is the embryonic shoot. This plumule consists of the embryonic leaves and the shoot apex, and it will develop into the stem and leaves of the future plant, growing upwards. Now, a key distinction for dicot seeds, which often causes confusion, is whether they are endospermic or non-endospermic. Endospermic seeds. In some dicot seeds, like castor, which gives castor oil, the endosperm, a nutritive tissue formed during double fertilization, persists as a prominent food-storing tissue, even in the mature seed. The cotyledons in these seeds are often thin, and the embryo utilizes this surrounding endosperm during germination. Non-endospermic seeds. In many other dicot plants like bean, gram, and pea, the endosperm is not present in the mature seed. Why? Because during seed development, the food reserves from the endosperm are completely absorbed and transferred into the cotyledons themselves. The cotyledons then become thick, fleshy, and the primary storage units.
So, in non-endospermic dicots, like our common pulses, little doll, the cotyledons are the primary storage units, having absorbed the endosperm earlier. That's a very important point for students to grasp clearly.
Precisely. It dictates where the food reserves are located in the mature seed. Now let's compare this with a monocotyledonous seed, which has some unique and distinct features. We'll use maize, corn, a staple grain, as our primary example. Please refer to Figure 5.15 in your study material for a detailed view. 8.3 structure of a monocotyledonous seed. As a general rule, most monocotyledonous seeds are endospermic, meaning they store their food primarily in the endosperm, and the cotyledon plays a different role. However, it's important to remember there are exceptions, such as orchids, which are non-endospermic monocots, but there are less common examples used in exams. Now, in the seeds of cereals, like maize or wheat, the seed coat and fruit wall have a unique relationship. The seed coat is very membranous and thin, and it is typically fused completely with the fruit wall, the pericarp.
Fused together, so you can't separate them.
Exactly. This fusion makes it difficult, if not impossible, to separate the two layers. That's why a maize grain, or a wheat grain, botanically speaking, is technically a type of fruit called a caryopsis, where the fruit wall and seed coat are inseparable. The endosperm in these monocot seeds is usually quite bulky and serves as the primary food storage tissue, much like in endospermic dicots such as castor. This large endosperm is what makes grains like maize, wheat, and rice so energy-rich and staple foods worldwide. Separating the outer part of the endosperm from the embryo is a distinct, thin, proteinous layer called the aileron layer. This layer is metabolically active and very important. During germination, it secretes digestive enzymes like amylases that break down the stored starch in the endosperm into sugars, making the food available to the growing embryo.
Aileron layer protein-rich makes enzymes.
Right. The embryo itself in monocots is typically small compared to the large endosperm, and it is situated in a groove at one end of the endosperm. It has some highly specialized components unique to monocots. It consists of one large, shield-shaped cotyledon, which is specifically known as the scutellum. This scutellum lies adjacent to the endosperm, and its crucial job is to absorb digested nutrients from the endosperm via enzymes from the aileron layer and efficiently transfer them to the growing embryo. It acts like an absorptive interface.
So the single cotyledon, scutellum, absorbs food rather than storing it like in many dicots.
Exactly. The embryo also has a short embryonal axis containing the plumule, embryonic shoot, and the radicle, embryonic root. But these are protected by special sheaths. The plumule is enclosed in a protective conical sheath called the coleoptile. Think of it as a protective cap that pioneers the way, allowing the delicate plumule to push through the soil during germination without damage. And the radicle is enclosed in an undifferentiated protective sheath called the coleorhiza. This protects the emerging radicle as it grows out.
Coleoptile protects the plumule, coleorhiza protects the radicle. Very specific monocot features. The presence of the scutellum, coleoptile, and coleorhiza are indeed very distinct identifying features of monocot seeds. This comparative study between dicot and monocot seeds is definitely a frequent question area in exams, so noting those precise differences, number of cotyledons, role of cotyledons, presence of endosperm, coleoptile, coleorhiza, is absolutely vital for scoring well. Hashtag EIX. Semi-technical description of a typical flowering plant. Right, so we've covered roots, stems, leaves, inflorescences, flowers, fruits, and seeds in quite some detail. To summarize and standardize all this vast morphological information, biologists use a precise, semi-technical description method. How is this actually done, and what exactly are these floral diagrams and floral formulas that seem so crucial for classification in exams?
This section is extremely important, as you say, especially for your practical examinations in biology, for higher-level identification in botany, and certainly for competitive exams, like NEET, where concise representation is key. It's all about presenting the morphological features of a flowering plant in a concise, scientific, and systematic way that allows for clear identification and easy comparison between different plant groups. 9.1 Description Sequence. The description always follows a specific logical sequence to ensure consistency and completeness. You don't just list things randomly. It typically starts with the plant's overall habit. Is it a small herb, a woody shrub, or a tall tree? What's its general form? This is followed by a detailed account of its vegetative characters, including the precise details of its roots, tap or fibrous, any modifications, stem, herbaceous or woody, erect or climbing, shape, surface, and leaves, simple or compound, phyllotaxy, venation, stipules. After thoroughly describing the vegetative parts, the description moves systematically to the floral characters. This covers the inflorescence type, racemose, cymose, etc., and then each individual flower part in a specific standardized order, always moving from the outermost whorl inwards. Calyx, corolla, androecium, gynoecium.
Habit, vegetative, root, stem, leaf, floral, inflorescence, flower parts, CCAG, a very structured approach.
Very structured, yes. And after describing all these various parts verbally, two highly standardized and symbol-based representations are presented to summarize the floral structure, a floral diagram and a floral formula. These are compact, universal ways to convey a lot of complex morphological information very efficiently. Let's break down the 9.2.1 floral formula symbols first. These symbols are universally recognized in botany and are absolutely critical for you to memorize for your exams. BAR stands for bractade, indicating the presence of bracts. If absent, sometimes EBR is used for bractade. K represents the calyx, the whorl of sepals. The number following K indicates the number of sepals. C represents the corolla, the whorl of petals. The number indicates the number of petals. P stands for perianth, used when the calyx and corolla are indistinct, composed of tepals. The number indicates the number of tepals. A represents the androecium, the male whorl. The number indicates the number of stamens. G represents the gynoecium, the female whorl. The number indicates the number of carpels.
BRKCPAG, with numbers for parts. Okay, what about fusion or ovary position?
Good question. That's encoded too. Fusion. If parts of the same whorl are united or fused, like gamosepalous or gamopetalous, this is indicated by enclosing the number figure within brackets or parentheses. For example, K5 means 5 sepals that are united. C5 means 5 united petals. G2 means 2 fused carpels, syncarpus. If they are free, polysepalous, polypetalous, epicarpus, there are no brackets around the number, if you equal K5, C5, G2. Adhesion. If floral parts from different whorls are united or adhered to each other, like stamens attached to petals, epipetalous, this is indicated by drawing a curved line or arc above the symbols of the floral parts involved. For instance, an arc connecting C and A, often written above them, indicates epipetalous stamens. For ovary position, G with an underline below it. G, this indicates a superior ovary, meaning the flower is hypogynous. The line is under the GG with an overline above it, G. This indicates an inferior ovary, meaning the flower is epigynous. The line is over the G with the line beside it. G, sometimes used for a half-inferior ovary, perigynous.
Underline G for superior, overline G for inferior.
I got it.
And for sexuality. A circle with a plus sign inside represents actinomorphic radial symmetry. Percent, a percentage sign or sometimes other symbols, represents zygomorphic bilateral symmetry, male symbol, denotes a staminate male flower, unisexual.
Right.
Female symbol denotes a pistillate female flower, unisexual. Bisexual symbol, like male and female combined, denotes a bisexual or hermaphrodite flower having both A and G. This symbol is often placed at the beginning of the formula.
Wow, a lot of info packed into symbols. Symmetry, sexuality, whorls, numbers, fusion, adhesion, ovary position. Exactly, it's a highly efficient code. Now let's talk about the 9.2.2 floral diagram. A floral diagram is a graphical top-down representation of a flower structure. If you refer to figure 5.16 in your text, you'll see a classic example showing both the floral diagram and the floral formula for the mustard plant, Brassica. This diagram provides rich visual information about the number of parts in each whorl, their arrangement within the whorls, e.g. aestivation, and the relation they have with one another specifically, cohesion, fusion within a whorl, and adhesion, fusion between different whorls. The position of the mother axis, the main stem or inflorescence axis from which the flower arises, is conventionally represented by a dot or a small circle on the top posterior side of the floral diagram. This orients the diagram correctly. The floral whorls are drawn as successive concentric circles, starting with the outermost calyx, sepals, then the corolla, petals, then the androecium, stamens, and finally the gynoecium, capillus, very cross-section, in the very center. Cohesion, fusion within a whorl, is shown by connecting the parts with lines or arcs within that circle. Adhesion, fusion between whorls, is shown by drawing radial lines connecting parts from different circles. For instance, figure 5.16 clearly shows the 4 free sepals, 4 free petals, 6 stamens, 4 long, 2 short, though the diagram might simplify this, and the central bicarpellary syncarpous ovary with parietal placentation, typical of the mustard family, Brassicaceae.
So the diagram is like a blueprint map of the flower from above. This concise symbolic language, both the formula and the diagram, is truly vital for quick reference in exams and in broader botanical studies, turning complex descriptions into easily digestible and comparable information. Absolutely. To really solidify our understanding of how all these morphological concepts and descriptive tools come together, let's apply them to a very important plant family mentioned in the material. Solanaceae. Why is it commonly called the potato family, and what are its key features according to our study material that make it so distinct and recognizable?
Phyllotaxy is indeed a large and incredibly economically important family, a great one to study as an example. It's commonly known as the potato family, simply because Solanum tuberosum, our common potato, is arguably its most prominent and economically significant member worldwide. But it includes many other familiar plants too. This family is remarkably adaptable and widely distributed across tropics, subtropics, and even temperate zones. You can refer to figure 5.57 in your study material, which visually illustrates Solanum nigrum, commonly known as McCoy, or black nightshade, another widespread member of this diverse family. Okay, let's break down its characteristics systematically, just like a formal description. First, the 10.2 vegetative characters. The plants in this family are mostly herbs, non-woody. Some are shrubs, and rarely do you find small trees. The stem is typically herbaceous, though it could be woody in shrubs, usually aerial, erect, cylindrical, often branched, and can be solid or hollow inside. The surface might be hairy, pubescent, or glabrous. And a very important modification we must mention again is the presence of an underground stem tuber in potato, Solanum tuberosum, specifically adapted for food storage. The leaves are generally alternate in their phyllotaxy, 1 leaf per node. They are typically simple, meaning the lamina is undivided or not incised to the midrib, though rarely they can be pinnately compound in some species. A crucial feature for identification. They are exstipulate, meaning they lack stipules at the base of the petiole, and their venation pattern is reticulate, typical for dicots.
Alternate, simple, exstipulate leaves with reticulate venation. Okay, those are key vegetative traits. Now for the flowers.
Right, the 10.3 floral characters. These are especially important for classification and distinguishing this family in exam questions. The inflorescence type can vary. Flowers might be solitary, borne singly, axillary, arising from the leaf axil, or more commonly arranged in a cymose type inflorescence, as frequently seen in the large genus Solanum, which includes potato, tomato, brinjal. The flower itself is typically bisexual, having both stamens and carpels, and actinomorphic, displaying radial symmetry can be cut equally through any central plane. The calyx consists of 5 sepals. Crucially, these sepals are united, gynocephalous, often forming a cup or tube. A notable feature is that the calyx is persistent, meaning it remains attached to the fruit even after it develops. Think of the green cap you see on a tomato or brinjal. That's the persistent calyx. The aestivation of the sepals, how they are arranged in the bud, is valvate, where margins just touch without overlapping. The corolla is composed of 5 petals, which are also united, gamopetalous, often forming a funnel shape, bell shape, or wheel shape that rotate. Like the calyx, its aestivation is also typically valvate. The androecium consists of 5 stamens. A critical feature for identifying Solanaceae is that these stamens are epipetalous, meaning they are attached directly to the petals, the corolla coupe.
5 united sepals, persistent, valvate. 5 united petals, valvate. 5 epipetalous stamens. Very regular so far.
Right.
What about the gynoecium?
The gynoecium is perhaps the most distinguishing feature of this family. It is bicarpellary, meaning it is made up of 2 carpels. These carpels are syncarpous, meaning they are fused together to form a single pistil. The ovary is superior, hypogynous flower. It is typically bilocular, having 2 chambers or locules. Now, 2 very characteristic features. The placenta is swollen, bearing many ovules, and the placentation type is axile, ovules attached to the central axis in each locule. Also, the ovary itself is often described as being obliquely placed within the flower, tilted slightly relative to the main axis, which is a subtle but important diagnostic feature for Solanaceae.
Bicarpellary, syncarpous, superior ovary, bilocular, swollen axile placenta, and obliquely placed. Lots of key details there.
Yes, those gynoecium details are vital. The fruits in Solanaceae are typically either a berry, like the fleshy tomato or brinjal, or sometimes a capsule, a dry fruit that splits open, like in datura. The seeds produced are generally numerous within the fruit, and they are endospermous, meaning the mature seed contains endosperm tissue serving as stored food for the embryo. Now let's summarize these floral characters concisely using the floral formula we learned. It would be, for actinomorphic symmetry, so for bisexual flower, K5, for calyx with 5 united sepals, C5 for corolla with 5 united petals, A5 for androecium with 5 stamens, and remember to draw the arc above C and A to show their epipetalous. And finally, G2 with an underline for gynoecium with 2 united carpels, and the underline signifying a superior ovary.
K5, C5, A5, G2, with the CA adhesion arc and the G underlined. That formula packs in all the essential floral info for Solanaceae.
It does. And lastly, the 10.4 economic importance of Solanaceae is vast, showcasing its huge impact on our daily lives. Many plants from this family serve as crucial food sources, including the highly popular tomato, brinjal, eggplant, and of course potato. Chili, a staple in Indian and global cuisine, is a widely used spice. The family also provides important medicinal plants like belladonna, source of atropine, and ashwagandha, Botany somnifera, used in traditional medicine. Tobacco, Nicotiana tabacum, used as a fumigatory and stimulant, also belongs here. And finally, many beautiful ornamental plants like petunia, browallia, and cestrum, night-blooming jasmine, are cherished members of the Solanaceae, adding beauty to our gardens.
Food, spice, medicine, fumigatory ornamentals, it really is a very important family. The Solanaceae is indeed a perfect example of how these precise morphological features we've discussed, from the vegetative parts to the intricate details captured in the floral formula, come together to define a distinct plant group and highlight its immense practical relevance in our daily lives. Hashtag outro roca.
So today, we've taken quite a comprehensive deep dive into the fascinating and fundamental world of plant morphology. We started by understanding the historical context and the profound importance of detailed observation and description in biology, a foundation meticulously laid by pioneers like Catherine Esau. We then systematically explored the structural organization of flowering plants, moving logically from the foundational root system, differentiating between tap, fibrous, and adventitious roots, and carefully dissecting the critical regions of the root tip, root cap, meristematic region, elongation, maturation, which are essential for growth and function. We moved upwards to the stem, identifying its distinguishing features like nodes, internodes, and buds, its primary functions of support and conduction, and the various ingenious modifications like tubers, tendrils, and thorns that allow plants to adapt to diverse conditions. Our journey continued to the leaf, the primary photosynthetic organ. We examined its general features, its 3 main parts: the leaf base, with potential stipules or pulvinus, the petiole and the lamina, and the crucial role of venation patterns, reticulate versus parallel, in classification. We then distinguished carefully between simple and compound leaves, pinnate versus palmate, noting the defining role of the axillary bud's presence or absence. And we explored the intricate patterns of phyllotaxy, alternate, opposite, and whorled, which optimize light capture.
A very logical progression through the vegetative parts.
Yes. Then we broadened our view to the arrangement of flowers, the inflorescence, defining it as a modified shoot and dissecting the 2 major types, racemose, with acropetal succession, and cymose, with basipetal succession, focusing on their distinct growth patterns. From there, we delved into the heart of angiosperm reproduction, the flower itself. We discussed its 4 whorls, calyx, corolla, androecium, gynoecium, categorized by function. We explored floral symmetry, actinomorphic, zygomorphic, and asymmetric, with clear, relatable examples. We understood how the position of floral parts on the thalamus defines the ovary position as superior, hypogynous, half-inferior, perigynous, or inferior, epigynous. And finally, we meticulously detailed each floral whorl, the protective calyx and attractive corolla, including the vital and often tricky concept of aestivation, valvate, twisted, imbricate, vexillary, the male androecium, its structure, filament, anther, cohesion patterns, mono, polyadelphous, and adhesion, epipetalous, epiphyllous, and the female gynoecium, its structure, ovary-style stigma, carpal fusion, alpal versus syncarpous, and the diverse types of placentation, marginal, axile, parietal, free central, basal. We followed this with the fruit, understanding it as a ripened ovary, its structure including the pericarp layers, epicarp, mesocarp, endocarp, exemplified by the drupe in mango and coconut,
And lastly, the seed, the plant's future generation, comparing the distinct structures of dicotyledonous, two cotyledons in endospermic, and monocotyledonous seeds, one cotyledon scutellum, usually endospermic, with coleoptile and coleorhiza. We concluded by seeing how all this detailed morphological information is synthesized into those useful semi-technical descriptions, concise floral formulae, and illustrative floral diagrams, as we saw demonstrated with the important Solanaceae family. This detailed descriptive biology isn't just about memorizing terms, it's truly about understanding the fundamental design principles of plant life. It forms the bedrock for, well, pretty much all other aspects of plant biology you'll study later.
That was an incredibly comprehensive deep dive, really covering Chapter 5 and the kind of depth needed for exam success. Thank you for that clarity. Now, for all of you listeners, to truly master this topic for your board exams and especially for NEET, we highly recommend you take some specific follow-up actions. First, definitely revise this topic again soon. While it's fresh, focus specifically on drawing and labeling all the diagrams mentioned, figures 5.1 through to 5.17 sun in your study material or notebook. Visualizing these structures yourself will really solidify your understanding and help you recall them under exam pressure. Second, make sure you practice identifying the different types of structures we discussed. Don't just read about them. Take any plant around you in your garden, on the roadside, and try to actively apply these terms. Is the venation reticulate or parallel? Is the phyllotaxy alternate or opposite? It makes learning active and much more engaging. Third, and crucially, solve past year questions, PYQs, based on this lesson. You absolutely must do this. Pay special attention to questions focusing on placentation types, aestivation types, the differences between monocot and dicot seeds, and of course the floral formula and characteristics of families like Solanaceae, as these are frequently tested areas and can be tricky if you haven't practiced. And finally, if you find you still have any doubts as you revise or practice these questions, please don't hesitate to revisit this deep dive session or reach out through appropriate channels for clarification. Remember, consistent practice and achieving clear conceptual understanding are your absolute keys to success in these competitive exams. Keep working at it.
And perhaps one final thought to leave you with as you reflect on this journey through plant morphology. Think about how these seemingly simple structural variations, a different type of root system allowing access to deep water, a unique pattern of leaf arrangement maximizing sunlight in a crowded forest, or the complex folding of petals in aestivation ensuring pollination by a specific insect, are actually incredible, ingenious adaptations. They represent nature's solutions, evolved over millions of years, allowing plants to survive, compete, and thrive in incredibly diverse and often challenging environments, from arid deserts to waterlogged swamps, from high mountains to salty coastlines. So ask yourself, how does understanding this intricate design influence our approach to modern agriculture, perhaps helping us breed better, more resilient crops that can cope with changing climates? Or how does it guide our conservation efforts as we work to protect and understand the immense biodiversity that depends on these plant structures? This knowledge isn't just for passing exams, it's truly a profound window into the genius of nature itself, and it has immense practical implications for our world and our future. Keep exploring, keep questioning, and keep observing the green world around you.